Welcome back. Okay, this lecture is basic explanation about the biochemistry and cell structure. Uh, it is, it may be very new for you. I have reviewed the biology course in your DME department and also in some other DME department. In the biology course, they usually teach whatever they teach. For example, if the researchers are focused on cardiology and eye, they focus on the biology of these two organs. But here, I want to explain some basic concepts for you. Just to become familiar, although it has, at first glance, it has nothing to do with bone and cartilage, but later, when we discuss the structure and different aspects of bone healing and cartilage healing, we will see in all the information. And also, if you become familiar with this term, it will be easier for you later if you want to think about some biomimetics. So, please forgive me for this session, but I think it is very necessary for someone who wants to work in the field and be related to medicine. Biochemistry is a subdivision of chemistry that studies the chemical reaction and chemical structure in living cells. And biochemistry has two sections, organic chemistry and inorganic chemistry. The inorganic part discusses the inorganic components that are inside the human body. When I'm talking about the human body, it includes all the animals, okay? And just to be simpler, I say human body. But they apply to all the mammals, at least all the mammals. The inorganic part forms the major component of our body. It contains water, salt, acid, and base. They are the medium for many reactions, not many, all the reactions happen in this inorganic medium. They are also important for many, not only they provide the medium, they are active in the chemical reactions, energy production, and also they provide the support for our body. For example, the inorganic salts constitute the hard section of the bone. If we lose this salt, the bone will be soft as cheese. Water constitutes the most part of our body. Almost 60 to 80% of our body is formed by water. And you are familiar with the properties of water. It has high heat capacity. It has a high heat of vaporization. You are familiar with them. I don't waste your time with explaining them. You know, the water molecule is a polar molecule. So these polarities help the water to solve any polar molecule. So it can be a solvent in our body that solves different materials and provides a medium for the reaction. Also, it works as a carrier to carry the nutrients towards the cell and remove the waste products from those cells. And uh, that is why we call water the universal solvent in our body. It is highly active, and also water provides some cushioning effects for our body. I explained in previous lecture about the cartilage structure, and I said that in those protoglycan brushes, there is water, and this water provides a cushioning effect. This is one function of the water. Another cushioning effect happens for your heart. Around the heart, there is a cavity that contains a little amount of water. But this water helps the heart to be kept safe inside the chest. If for example, when you are running, your heart is moving while you are jumping up and down during the water run. But the cushioning effect of the water holds your heart in place. Another part that water functions as the cushion is the fluid around the brain. Actually, our brain is floating inside a cavity which is filled with fluid. First, this floating condition decreases the weight of the brain, so the brain can be held in place easily. And if something hits the head, this trauma will not be transferred directly to the brain. It will dissipate in the fluid around the tissue of the brain. So in this way, the brain and the spinal cord are kept safe inside cavity of water. So water is very important for our body. Salts. The definition of salt is an ionic compound that contains cation and anion, but the cation and anions are must not be hydrogen and hydroxy group. Anything other than hydrogen and hydroxy, like NaCl or KCl, they are salts, and they easily dissolve in water because they are ionic and water is polar. So they dissolve in water and dissociate into their into their ions. The major salt in our body is sodium chloride, the salt you eat with your food, calcium carbonate, calcium phosphate, and potassium chloride. There are many other salts in our body, but these are the most important ones. Calcium chloride and potassium chloride are important for cell function and nervous system function and cardiac function. Calcium carbonate is important for balancing the acidity in our body, and calcium phosphate is the most abundant salt in our body, and this is the salt that forms our bones and teeth. And the hardness of our bones and teeth is dependent on the calcium phosphate. Again, you are familiar with the definition of acid and base. Acid is a chemical that releases hydrogen ions when dissolved in water, and base is chemical that releases hydroxide ions. The same definition applies in the nucleic acid in our body, but not only the organic acids are present in our body, we have some, not only inorganic acids are present, we have some organic acids. The organic acid, I'm sure you are familiar with the inorganic acid formula. When you are writing the formula for chemical formula for an inorganic acid, you always write the H at the beginning, and any formula that begins with H is an acid, like H2CO3 or HCl. These are acids. These are inorganic acids. But organic acids are not written like that. And based on the chemical formula, you cannot say it is an acid. You must know that it is an acid. Lactic acid, citric acid. Acid, bases are also the same. For example, some bases are inorganic, like carbonate. You know, when we have a weak acid and it falls in water and it dissociates into ions, for example, H2CO3 dissociates into bicarbonate, HCO3 negative, and hydrogen ion. Now, if you look on the other side of the reaction, this bicarbonate, when solved in water, it dissolves the hydrogen ion, so it is acting like a base. So any weak acid, when dissolved in water, from the acid side, it is an acid. From the other side, it is a base. One of the most important acids in our body is carbonic acid, but more important than the acid is its base. It is the most important base controlling the acid-base balance in our body. Acid-base balance is very important for our body. The pH of the fluid inside our body is 7.324, and it must be kept strictly in this range. If it changes a little bit, many functions will be lost. So it's important to keep the acid-base balance, and one of the most important molecules that does this is bicarbonate. Inside these famous acids and bases, many other molecules, even large molecules in our body are either acid or base, like amino acids, fatty acids, proteins are acid. Or DNA and RNA, which are large molecules in our nucleus, in our cell nuclei, they are base. So we have lots of acids and bases in our body, but the balance between the acid and base is dependent on this vitality. So when we discuss the organic compounds, we are talking about a molecule that contains carbon and hydrogen at the same time. The only exception is carbonic acid. Carbonic acid can be considered an inorganic one, but you can also consider it organic. Any other molecule that contains carbon and hydrogen at the same time is organic compound. There is no exception. The biology on Earth depends on carbon. It's based on carbon. Carbon is a unique atom. You remember the periodic table? It is located in the fourth group, just in the middle of the periodic table. It has both the capabilities of the atoms in the its left and also on its right. So it can interact with both of those atoms and also with itself. It has 4 electrons, has valence electron, and it can share all these 4 atoms. So it can produce 4 bonds with 4 other atoms. These 4 other atoms can be any atom, including the carbon itself. And this property helps carbon to create very large molecules containing several carbons attached together. When we have an organic molecule and it wants to attach to another one, almost always, it happens through a chemical reaction that includes water. And it is called dehydration. As you see here, 2 molecules want to attach together. Hydroxyl group from one molecule and hydrogen ion from the other molecule detach from these 2 molecules and join together and form a water molecule. And then these 2 molecules join and form a bond between them. This reaction, which creates larger molecules and 1 molecule of water, is called dehydration. If we reverse it, we break the bond here and add 1 hydrogen group and 1 hydroxyl group to each of these molecules. We can separate them with use of 1 water molecule, and this reaction is called hydrolysis. So dehydration is the opposite of hydrolysis. In dehydration, 1 molecule of water is created and a larger molecule of the original material is formed. In hydrolysis, it is a breaking reaction. It breaks the larger molecules into smaller ones and uses 1 molecule of water. Almost all reactions in organic material happen by these 2 reactions. Now, let's review very fast the organic material. The first one is carbohydrate. You are familiar with carbohydrate. You will use sugar every day. Sugar is a carbohydrate. Carbohydrate, we have 3 major chemicals in our body. Carbohydrate, lipid, and protein. Carbohydrate constitutes 1 to 2% of our cell mass, but it is very important because it is the major energy supply, not energy reserve, energy supply for our cells. Our cells cannot use energy in any other form other than glucose. Glucose is one of the carbohydrates, one of the simple carbohydrates, and everything we eat must be turned into glucose inside our body to be able to be used by our cells. There are some exceptions. Sometimes the lipids, proteins are used directly, but they are special conditions. Under normal condition, everything you eat gradually turns into glucose and your cells use glucose. So without glucose, your body doesn't function. If you have one molecule of carbohydrate, it is called monosaccharide. If you have two molecules attached together, it is called disaccharide. And if it is a polymer, it is called polysaccharide. The monosaccharides have a simple chemical formula. They are formed by carbon, hydrogen, and oxygen. They don't have any other atom. The ratio between these three is 1:1. For one carbon atom, we have two hydrogens and one oxygen. So based on the number of carbon, we name them. Usually in our body, we have two kinds of monosaccharides. We have monosaccharides with 5 carbons or with 6 carbons. The 6 carbons are used for energy, and 5 carbon monosaccharides, which are called pentose, are used in DNA and RNA structure. These are disaccharides are common in our body, but when we want to use them, they must be broken into their components. Polysaccharides are in two sorts. One is the starch. The starch is not familiar. We use the starch for cooking and baking. Starch is produced by plants, and we eat the starch in our gastrointestinal system. The starch, which contains, which is a polymer of glucose, is broken into smaller parts, and these smaller parts are absorbed and turned into glucose and used. But in our body, we produce another polymer, which is called glycogen. Glycogen is the same as a starch, with the same structure, with more branching, but since it is produced in our body, in animal body, we call it glycogen, and the other one is starch because it is called, it is produced by plants. The difference is just the origin. And glycogen is the storage form of glucose in our body. Whenever we have excess glucose, it polymerizes and turns into glycogen and is kept in a safe place in our body, inside the cell, like inside the muscle, in the liver. And whenever we need them, it is released and we can use the glucose. So glucose, carbohydrate is the major energy source for our cells under normal conditions, and also it is a major part of cell structure, specifically for the surface of the cell, which helps in signaling between the cells. The next organic chemical component in our body is lipid. You are familiar with lipid. Lipid is the oil we use for cooking. They have several types, but the most important thing about them is they are insoluble in water, so they cannot dissipate in our body, but they can dissolve in other lipids. And wherever we have lipids, they can penetrate, and also they can be dissolved in alcohol and ether. They have several types. The most common one is called triglyceride. The name shows there must be 3 of something. They have a common structure, all of them. We have maybe countless number of triglycerides, but they all have a common structure. There is a small molecule with three carbons called glycerol. It is common among all triglycerides. It is the same molecule. The second part is a large molecule with a lot of carbon called fatty acid. This glycerol is an alcohol. It has three hydroxyl groups. The fatty acid can attach to the hydroxyl group. So three fatty acids can attach to one single glycerol, and it creates triglyceride through a dehydration reaction. So we have a large molecule, which is a liquid called triglyceride, and it is insoluble in water. If all the bonds between the carbon in the fatty acid are single bonds, it is called saturated lipid. Saturated lipid is solid at room temperature. For example, the butter is solid at room temperature. So it is saturated oil or liquid. But if there is a double bond between carbon, if there is one single double bond, it is when there is double bond, they will be unsaturated. And based on the number, we call them monounsaturated with one double bond or polyunsaturated with more than one. When they are unsaturated, they are liquid at room temperature. For example, olive oil is unsaturated, and it is a polyunsaturated triglyceride because it is liquid at room temperature. Or other like canola oil is also polyunsaturated. What is the function of lipid? The function of triglyceride. These are the primary source for energy storage. When we want to store energy for later use, we turn it into triglyceride. But I said the primary source for energy use and energy supply is carbohydrate. I'm insisting not to make a mistake, but just I want you to know. Another type of lipid, which is very important in the structure of our cell, is called phosphorus. It will remove one fatty acid from the glycerol and add a phosphate group instead of the one fatty acid. It is called phospholipid. It has a phosphate group, glycerol molecule, and two fatty acid molecules. And since the phospholipid has a specific structure, it has a unique structure with one double bond. It's always shown as this shape you see, with a bending in one leg. It is unique if you remember here, everything is hydrophobic. So this is insoluble in water. But in phospholipid, the phosphate group is hydrophilic. It likes water and it can dissolve in water. But the fatty acids are hydrophobic. They repel water. So they are insoluble in water. So this is a bipolar molecule. One side is soluble in water, insoluble in water. The other side is soluble in water. And it is a unique property that we will later see how it is important because it is the basis for creating the cell membrane. Another form of lipid is called steroid. You are familiar with the most common type of steroid. It is called cholesterol. I'm sure you have heard about cholesterol, about the bad cholesterol, good cholesterol. It's harmful for the body, it's less cholesterol, but cholesterol is a necessary part of our body. It is steroids are specific type of lipids which have structure with four rings. And the basic and the major fluid in our body is cholesterol. Cholesterol is important because it forms the part of the cell structure, the cell membrane structure, the wall of the cell. It is the precursor to many hormones and chemicals in our body. And also, you know, if you are under sunshine, your body starts to produce vitamin D. The precursor for vitamin D is cholesterol. So this is a medical necessity to have cholesterol in our diet. And it's very important to our body. But the most important structure, chemical in our body is protein. If you lose your lipid, you will survive for several weeks. If you lose your carbohydrate, you will survive for several days. But if the proteins in your body are destroyed, you will die immediately. The body cannot survive without protein because they perform the major and vital functions in our body. They form 10 to 30% of our cell mass, so they are a major part of our body. They are complex molecules. They are formed by a building block called amino acids. These amino acids are connected together by a dehydration reaction and form the peptide bond. The amino acid has a unique structure. We have 20 different amino acids in our body, and all the proteins in our body are formed by these 20 different amino acids. Their structure includes a central carbon, which is called alpha carbon. It is attached to 4 different groups. One is an amino group, one is a hydrogen, the other one is carboxyl group, and these 3 with the alpha carbon are the same in all these 20 different amino acids. The fourth connection is to an R group. This R group is different among these 20 amino acids, and this is why we have to do this about it. It can be simple like a hydrogen, or it can be a complex group. But this is the structure of amino acid. When the amino acid wants to join, the hydrogen from the amine part and the carboxyl group from the acid part, the carboxyl group, are attached and joined, together to form a water molecule, and the amino acid join and form a peptide bond. And it can happen on the other side of each amino acid, so they can create a large chain of amino acids. The simple chain of amino acids forms the protein. The number of amino acids and the location of each amino acid determines the type and properties of the protein. But this is not that simple. Unlike the carbohydrates and lipids that we didn't discuss their three-dimensional structure because their properties is not dependent on their two-dimensional structure. The proteins are dependent on their three-dimensional structure. This primary structure is the chain structure of the amino acid, but these chains form helix or sheets, and these sheets can, along their length, can acquire different shapes, as you see, and these large molecules of containing polymers of amino acids can join together and form a large protein molecule. Not only the arrangement of amino acids determines the properties of the final molecule, the secondary, tertiary, and quaternary structure of proteins also determines the function. And if each of them is disrupted, the protein no longer functions. So to destroy a protein, you don't need to disrupt this peptide bond. We just need to disrupt this structure, and it is much easier to do that. Every time you put an egg and eat it, you are destroying the protein inside the white of the egg. In the raw egg, the white is not white. It is transparent. Yes, it is like water. But when you cook it, you boil it, you fry it, it turns into a white, non-transparent material. Yes, the white of, then you call it the white of egg or egg white. Actually, you are not destroying, you are changing the protein structure in the egg white. This process is called denaturation. And this denaturation that you do on egg is good because if you don't do that, you cannot eat it. But if it happens in the on the protein in our body, it is dangerous. One function of the protein is to facilitate the chemical reactions in our body. In chemistry, you are familiar with catalysts. Catalysts increase the chemical reaction speed and decrease the activation energy necessary for that reaction. Enzymes are a kind of protein that act as catalysts in our body, but they are biology catalysts. They decrease the activation energy for the chemical reaction, so the chemical reaction can happen much easier. How they do it? This is an enzyme, and each enzyme is specific for a specific substrate and specific chemical reaction. So we have lots of enzymes in our body. Each for one chemical reaction and for one substrate. The enzyme has some sites called active sites that can attach to the substrate. Without the enzyme, these substrates can react to each other, but they need lots of energy to, to stop the chemical reaction. But without any energy used, they attach to the active site on the enzyme. Once they are attached, the enzyme undergoes configurational changes. The 3D structure of the enzyme changes in a way that brings these two substrates close together, so close that they can now react with each other. They can undergo a dehydration process, any process that they need, and then they produce a product. And once the product is produced, they are released, and the enzyme returns to its original shape. The next molecule in our body is nucleic acid. Nucleic acid is the molecule of life. Our life, all the living creatures on the earth are alive because of DNA. I'm not talking about the soul and the other part. I'm talking about the biology, okay? Without DNA, there would be no life on Earth. DNA, sorry, not DNA, nucleic acid, because DNA is a kind of. Nucleic acid is the largest molecule that we ever have known. I mean the biology molecule, not the non-biology. Sometimes it is about 2 or 3 meters in length. And it is inside the nucleus of each cell. In each cell, we have 46 chromosomes inside the nucleus. Each chromosome is made of one single strand of nucleic acid, and this nucleic acid forms the basis for all the information that the cell and our body needs for living. We have two major types of nucleic acid. One is called DNA, deoxyribonucleic acid. It is a double-stranded molecule, and it is helical. It is the genetic library of our cells. The other one is ribonucleic acid, or RNA. It is single-stranded, but the structure is almost similar. The building blocks of these huge molecules are 4 building blocks, and these 4 building blocks, all the genetic information are provided. Each one of these building blocks is called a nucleotide. Each nucleotide contains one phosphate group and one pentose sugar, pentose carbohydrate. You remember I said carbohydrates have 6 carbons or 5 carbons. The 6 carbons are used for energy, and 5 carbons are inside the nucleic acid. This is that 5 carbons. It can be deoxyribose or ribose based on this sugar. The name is different. So in DNA we have deoxyribose. In RNA we have ribose. And the other end of the sugar, the sample sugar, is attached to a base. There can be 5 bases: adenine, guanine, cytosine, thymine, and uracil. Based on these bases, you have 5 different nucleotides. 4 of them, adenine, guanine, cytosine, and thymine, are present in DNA, and they form DNA. RNA doesn't contain thymine. Instead, it has uracil. So there are 3 differences between RNA and DNA. One, RNA is single-stranded, DNA is double-stranded. RNA contains ribose as pentose sugar, DNA contains deoxyribonucleic. And the third difference is RNA contains uracil, DNA contains thymine. You see only a section of a DNA. It is very huge and large, and it contains millions of these building blocks attached together. Each 3 of these building blocks are the code for one amino acid. The only thing they do, they code for one amino acid, each 3 of these building blocks. For example, GAT is for one amino acid, PCT is another amino. So they code for synthesis of different proteins, and these proteins are the basis for all the functions in our cell. The whole length of the DNA contains several smaller segments. They are not separated, they are continuous, but they are functionally separate. Each one of these functionally separated segments is called a gene. So gene is a section of DNA. Each DNA or each chromosome contains maybe several thousand, several hundred to several thousand genes. And each gene is responsible for one protein. Each gene contains many of the building blocks. So based on the number of amino acids necessary for each protein, you have the same number of the 3 codes for the amino acid. This is a chromosome, and this is the nucleus of a cell, and the DNA and chromosomes are inside the nucleus. And when we need them, they separate. They are because they are very long, they have to be formed in a helix, and they fold over and over to be able to reside inside the tiniest space of the nucleus. But whenever we need a protein, these molecules unfold and code for a protein. The last molecule we are going to discuss is another nucleotide. I said nucleotide is the building block of nucleic acid, DNA and RNA. But we have another nucleotide that is not part of the DNA and RNA, and it is called adenosine triphosphate, or ATP. ATP is much more famous than adenosine triphosphate. If you say your division adenosine triphosphate, you may think for one to remember what it was. But ATP, everybody knows what. ATP is a free form of the nucleotide, which is contains instead of one phosphate. If you remember here, we have one phosphate, one pentose, and one base. Here we have one base, which is adenine, one pentose, which is ribose. But instead of one phosphate, we can have two or three phosphates. With one phosphate, it is called adenosine monophosphate. With two adenosine diphosphate, and then adenosine triphosphate. AMP, ATP, and ATP. These molecules, ATP, is the energy currency in our cells. When you work and you earn, you will earn by dollar. They give you some bill at dollar, and whenever you want to buy something, you spend this dollar. So dollar is your currency for your earning. In our body, chemical reactions happen. They use glucose and break the glucose into water and CO2. So the energy inside the glucose is released, but this happens inside a part of the cell, and this energy is released there, but we may need the energy in another part of the cell. How this energy can be transferred from one part of the cell to the other part to be used in another reaction. This is the function of ATP. As you see here, when the second phosphate wants to attach to the first phosphate, due to their three-dimensional configuration, attachment of the second one to the first one is very difficult. So it absorbs a lot of energy to attach. So it creates a high energy bond here. And when released, when this bond is broken, a lot of energy is released. The same thing happens between the third and second, but imagine now the attachment of the third due to the three-dimensional configuration is much more difficult. So the energy for the second bond is larger than the first one. So here we have two large energy bonds that store a lot of energy, and when released, they can release the huge amount of energy. So when we are, our body is breaking down the glucose and produce energy, it's producing energy. This energy, which is released, is stored as ATP. Then the ATP as a molecule can be transferred to anywhere that the cell needs, to the other part, and then at that part, the phosphate bond is released and the energy is released, and the energy is used by that part. Then the ATP is turned into ADP or AMP, usually into ADP. Our body doesn't want to break the second one. It always wants to use the first one. So the ATP then returns to the original energy source and captures another phosphate and then returns. So this recycling process happens always in the cell. Any questions? I'm sure this discussion is interesting for you, but this is you. But in future, you will see how effectively this knowledge. Now we discuss all the molecules that construct the cell. Now let's see what is the structure of the cell. How these molecules gather together to form a cell. A cell is the smallest independently functioning unit of an organism. Each cell has separate, different structure inside it, but those structures cannot function independently. So one cell can function independently. You can remove one cell of your skin, not the dead cell on the skin, from the base layer, one cell, and put it in a growth stadium, and under correct condition, it can grow. Each cell is enclosed inside a membrane. This membrane is called plasma membrane, and it separates the external environment from the internal environment. And also it contains genetic material, which is based on the cell. It can be DNA or RNA. This genetic material encodes for all the functions that the cell needs to perform. How? By coding for protein, and these codes informs the specific part of the cell to create new protein, and those proteins are responsible for that specific function. That function can be anything, can be a reaction, can be structural function, can be signaling. The cells can be, generally speaking, the cells can be a single, an organism can be a single cell, like a bacteria, or can be complex, like a human and animal and plant. When we are talking about the cells, we have two types of cells in the nature on the earth. One cell is the preliminary form of life, which is called prokaryotic. It is the form that we see in bacteria. This is the original shape of life. The second one is eukaryotic. True. Karyon means nucleus, eu means true, so true nucleus. Why we call them true nucleus or eukaryotic? Because these cells, like all cells, contain a true nucleus in their structure. Actually, all cells, which are all eukaryotic cells, contain three separate structures. One, a plasma membrane surrounding the cell, a nucleus inside the cell that contains the genetic material, and everything that is between these two is called cytoplasm. In eukaryotic cells, there is a nucleus with a membrane that contains the whole genetic material inside it. But in prokaryotic cells, they don't have any nucleus. The nucleic acid and the nuclear material are dispersed inside the cell. Let's review very fast the structure of eukaryotic cells. As I said, we have a plasma membrane in the periphery separating the external environment from internal environment of the cell. We have a nucleus in the, not in the middle, inside. It can be anywhere in the cell. And anything between this nucleus and the plasma membrane is called cytoplasm. Cytoplasm is the machinery of the cell. Lots of organelles or small organs are inside this cytoplasm. First, let's discuss briefly the plasma membrane. You remember phospholipid. It has two fatty acids, one glycerol, and one phosphate group. And as you remember, it had two legs, these were fatty acids, one leg was fat. Since the phosphate is hydrophilic and fatty acids are hydrophobic, it created a bipolar molecule. Now let's see how it works. All the fluids in our body are water-based. So this is outside the cell, this is inside the cell. Inside the cell contains water. Outside the cell contains water. So the phospholipid wants to create a membrane. The phosphate group faces toward the outside. The tail, which are hydrophobic, cannot face the inside because here is again water. So another layer of phospholipid is formed inside with the phosphate group facing the water. And this forms a bilayer of phospholipid with the phosphate group facing outward toward the water, which is outside and inside the cell, and the tails facing toward each other. This whole thing is the plasma membrane. This is one membrane, but it's formed by two layers of phospholipid. All the membranes in our body, inside the cell, outside the cell, everywhere, when we have a membrane, this is the structure. Phospholipid bilayer. And since the center of this bilayer is lipid and it is hydrophobic, the lipids can dissolve in this membrane and pass through. But this is not the only component in the plasma membrane. Plasma membrane contains several proteins. It contains cholesterol. You remember when I discussed the cholesterol, I said it is not only for clotting the blood vessels, it is part of the cell structure. And you see it is present in the plasma membrane. And we have carbohydrates on the outer surface of the membrane, which are used for signaling and for identifying the cell. Except for blood cells that are free to move, every other cell in our body is attached to something, attached to a base or attached to another cell. Each attachment is important for their function. We have several junctions between cells, several types of junctions that provide the stability to cells. One of the problems that we have in recreating the tissues is we are not able to recreate these junctions. One of them is tight junctions. The name shows there are very tight attachments between cells. These attachment does not allow the passage of anything between the cells. So it seals the space between the cells. Another one is desmosomes. Desmosomes are like Velcro tape. You are familiar with Velcro tape. These desmosomes have some attachments inside the cell, but between the cells there are some proteins that connect to each other, but the space is not occluded. Water can pass through it. Small molecules can pass. Exactly like a well-closed can. Okay. Uh, so when we have desmosol, the space between the cells are attached together. The cells are attached together, but the space between the cells are not occluded. And the third type is gap junction. There are some channels attaching two cells together. So the inside environment of these cells and the inside environment of the adjacent cells are connected through this channel. So if there is any charge inside one cell, it can move to the other one. If there is any molecule, a small molecule, it can move to the other one. You will see later in muscles and nerves, we use these junctions for communication between the cells. And as I said before, let me explain the nucleus first. The nucleus is like a small cell. It has the same membrane we discussed, the same structure. And this membrane covers the whole genetic material and all the DNA inside it. In the center, we have small nucleus called nucleolus. Small nucleus. So this is the whole nucleus, and it contains all the genetic material that is necessary for the function of our cell. So now between the nucleus, which is the hallmark of eukaryotic cells, and the plasma membrane, we have cytoplasm. Cytoplasm contains several organs. I just review them very fast for you to become familiar. I don't want to review to go deeper. We have mitochondria, which is the site where the glucose is broken down into water and CO2 and the energy is released. So mitochondria is the power source of the power plant for our cells. There are very tiny organelles inside the cytoplasm. They are called ribosomes. They are responsible for synthesis of protein. When the gene codes for a protein, this code produces an RNA from the DNA in the chromosome, and RNA is produced. The RNA contains the code from the gene, so it has almost the same length of the gene, not the whole protein. It moves from the nucleus to the cytoplasm. In cytoplasm, this RNA attaches to the ribosome, and the ribosome decodes the code on the RNA and turns those codes into amino acid attachment together. So based on the code, the ribosomes move on the RNA, and different amino acids are attached together, and a protein is formed. This process is ribosomes, and the protein synthesis happens in an organelle called endoplasmic reticulum. The blue one is here around the nucleus. So all the proteins that are produced in our cells are produced in the cavity, and they collect these proteins and release them as something called heavy global part that contains the membrane and contains the products of the protein, the produced protein. They can release these proteins on the surface, as you see here, or they can transfer them to another part, which is called Golgi apparatus. It is responsible for post-production process. So the raw protein here is carried to another part, which performs post-processing to make it as the final step. We have another section here called a smooth endoplasmic reticulum. It is responsible for storing some material inside the cell. You need to be familiar with it because it is a major part in the skeletal muscle cells that store the calcium, which is vital for skeletal muscle function. The calcium inside the skeletal muscle function is stored in smooth endoplasmic reticulum. Inside the cells, we have some of these vesicles that leave the Golgi apparatus become lysosomes, which are the digesting machine for cells. If they bite anything, they can digest it. So if there is any injured or damaged part in the cell, they digest it. And also we have lots of fibers, tubules, and microtubules inside the cell, which is called cytoskeleton. There are several types. We don't want to discuss them. On the surface of the cell, based on the type of the cell, we have some projections. They can be single or multiple. This is the whole structure of a cell. Not all the cells have the same structure, and the number of each one of these parts may be different in different cells based on their needs. For example, the fat cell doesn't need to produce protein. So the fat cells, as you see, they are white because they contain large vesicles of fat. The vesicles in fat cells are huge, large, and numerous. Or in liver cells, we give lots of these rough endoplasmic reticulum. So those cells in the liver have a huge number of endoplasmic reticulum. So based on the cells, they are different. Their structure may be different, but the basic structure is the same. Our cells, your cells, the cells in your body, and the cells in my body are not divided. They are resting. They are living. Except for some cells like the blood cells or the skin cells that they must be renewed every day. The other cells have been developed during the infancy, and after that, they are in the resting phase and they are living their life. But sometimes some of our cells, like the blood cells, the skin cells, the cells inside covering the mouth, covering the gastrointestinal system, they are damaged every day and they need to be renewed. And also the cells in the genital system, they need to divide and renew and create new cells and ovum. So once it is necessary, they leave the resting phase and enter the division phase. The division phase or cell cycle has two parts. One part is the part is filtratory part, or we call it interface. During this phase, the cell begins to grow. The genetic material which must be divided between the cell, two daughter cells, the genetic material will be duplicated, and then the cell is ready for, and all the organelles are duplicated, so the cell is ready to enter the division phase, from interphase to division phase. When the cell enters the division phase, the cell begins to divide the genetic material into two identical groups, and then the cell divides into two identical cells. That is how our genetic material are transferred to the next generation of cells. This is the cell cycle. Not all the cells enter these cycles. For example, the nerve cells, the muscle cells, they never enter this. And the other cells are also in resting phase. For example, your liver cells are in resting phase. But if anything happens to those cells, they can leave the resting phase and enter the cell cycle and create new cells to replace the damaged cells in your liver or in your eye, in your lung. This is what happens during a cell cycle. You have an interphase and the division phase, which is called mitosis. Mitosis is non-sexual division. The number of chromosomes between the mother cell and the daughter cell are the same. This is for non-sexual division. The liver cell, the lung cell, or any cell in our body, except the genital. We have another division, just I mentioned it because you become familiar with it, but you don't need to know more about it. It is called meiosis. This non-sexual division is mitosis. The sexual division is called meiosis. And that is a specific division happening only in testes, in male, and in ovary in female, to produce the sexual cells for conception. And that is a different story that is not related to the specific cells. So whatever we discuss about the prokaryotic cell, eukaryotic cells. The cells give a true nucleus. But the origin of life on Earth started with prokaryotic cells, the cells that do not contain a definite nucleus. These are simple cells, but our life depends on them. We cannot live without bacteria. If you like yogurt, if you like cheese, if you like if you like bread, you cannot have these without bacteria. But they are good bacteria. Most of the function of digestion in your large intestine happens by the help of bacteria, but good bacteria. The surface of your skin are covered with bacteria. And if they are not there, you will become sick because they prevent the bad bacteria to enter your skin, to invade your skin. So these are good bacteria, and we depend on them. We cannot live without them. Our problem is with bad bacteria. And bad bacteria are only 1% of all bacteria. But the produce significant problems. Bacteria are simple cells. They usually as a single organism. They can have different shapes, like a sphere, rod, radio, asteroid, or a spirochete, you see different shapes. But the most common ones are these two. These are very specific kinds of bacteria. The most common ones are spherical or cylindrical. The cylindrical one is called bacillus. The spherical one is called bacillus. And you see these are retained. They are much smaller than the human cell. They are usually from upper micrometer, upper micrometer, 2 to 5 micrometers. They can live as a single organism, as a single cell, or they can attach together to form a chain, a tube, or a bunch of bacteria, but it doesn't change. When they attach together, they will not become an organism with multiple cells. Each cell is functioning and living separate. They are not dependent on each other. They just attach to each other. Let me go back to this shape. This is the simplified shape of a eukaryotic cell. And this is the simplified shape of the prokaryotic cell. And you see how simple they are. They have a cell wall, which covers the plasma membrane. This cell wall is specific for bacteria, and most of the function of bacteria, their pathogenicity, their survival in different environment, their state, everything is dependent on this cell wall. And it has a complex structure. Then under the cell wall, they have plasma membrane, and then they have cytoplasm. Inside the cytoplasm, because they need some proteins to be synthesized, they have some ribosomes. There's few ribosomes. And instead of nucleus, they have a nucleus area, a nuclear area. This area contains the nucleic acid, and you see there is no nucleus. The DNA or RNA is dispersed inside the cytoplasm. The glues in bacteria is different from the glues we discussed, the cell cycle and everything that happens. When we put the bacteria on a medium to glue, wherever it is, in the ocean or in the lab, first they pass through a phase called lag phase. They do not glue. They try to assess the environment to see how they can glue. And then when they assess the environment, they start to divide into two cells. One cell divides into two. Each one of those two cells divides into another two. So they divide in a binary region. So the growth rate will be exponential. So they divide very fast. So they grew in number very fast until they reach the plateau. Why they reach the plateau? They grew no more because the supply of nutrients in the environment is limited. So if they grow more, They don't have enough nutrients, so they will die. That is why they stop to grow and they continue to use the material in the environment. As long as the nutrients in the environment are present, they continue to stay in this state. But as soon as their nutrients are depleted, they start to die and the number of bacteria decreases. I'm discussing these parts of the bacterial life because one part, one important part in orthopedic surgery is prevention of infection. Why infection is so important in orthopedic surgery? Because of two problems. One is the structure of the bone. Bone has a porous structure. There are many holes inside the bone, microscopic and macroscopic. When there is an infection, the bacteria can go inside those holes, and we have no access to those bacteria to remove them by surgery. And also, since the bone doesn't have a good blood supply, we cannot reach the antibiotics to those bacteria that are residing inside the holes inside the bone. So the bacteria will remain in the bone and grow and destroy the bone. This is one problem. The second problem is the implants and the devices we use. All the surgery, orthopedic surgeries are associated with one device, one something. This something is an external, and it is something that comes from outside the body. So it is foreign body. The bacteria like to attach to the surface of metals of foreign body, and when they attach, they secrete a layer of material that is called biofilm. This biofilm covers the bacteria attached to the surface of the implant. And it protects the bacteria from antibiotics and from any surgery that you do to wash the bacteria away. We cannot wash this biofilm. When we wash the implant, the biofilm is attached there and we cannot remove the bacteria. So the bacteria remains there. Antibiotics has no effect on it. Surgical washing has no effect on it. It is attached to the surface of the implant under the biofilm and it remains there as long as it wants. So due to these two reasons, whatever we do, we must prevent it from happening. Biofilm formation is a known phenomenon for bacteria. In anywhere that we have bacteria and the metal surface, we have biofilm. Over the surface of the sheets and containers in the ocean, they are all covered by biofilm containing the bacteria inside the ocean. In many factories, for example, in pasteurizing some factories, pasteurization of the milk, they are made by stainless steel. All the devices are made by stainless steel, and this is a metal surface. After a while, all the surfaces are covered with bacteria, with biofilm, and they have to wash it, use specific cleaning strategies to remove these biofilm. So biofilm is a problem for humanity, but specifically in orthopedic surgery, it is catastrophic because when infection happens in orthopedic surgery in a patient with implant in the body, it remains there forever, almost forever. There is some surgeons sometimes say when you have an infection, infected patient, you have two ways to get rid of that patient, either kill the patient or cure it. Otherwise, the patient will survive as long as the infection will survive as long as the patient and physician survive. That is why it's important to know, to be familiar with the bacteria, how they work, and whatever we design, design something to prevent this infection. You saw in the previous lecture about the cell-based purification. That was a genius idea. I mentioned some problems with that, but the idea is genius. And if we can do something to prevent the biofilm formation, because if there is no biofilm, we can wash the bacteria away from the surface of the infection during the therapy. But with biofilm, we cannot wash it. We have to scrape it and it is not possible because sometimes the implant is inside the bone and you cannot reach it, you cannot remove it. If you remove it, the surgery is ruined. So that is why I think it's hard for you to be familiar with the concept. Okay? Any questions? Next session will be on Monday. It is about the research. I just request you to review the research title I gave you in the syllabus for the research, the applied orthopedic engineer. Okay? If you open the syllabus, I have written several titles in red in the syllabus for applied orthopedic engineer. Review those titles, you must select one of them for your research during this year, okay? On Monday, I will explain those titles and I will explain everything for you so you have a better idea for selecting your research. But just review those research before to be familiar with the title, okay? So see you on Monday at 6:30. Have a great weekend.
Thank you. Have a good weekend.